Okay, so now we're on Microbial Evolution, Chapter 15. This is video number 2. So you should have already filled out part of your study guide. If you look at your study guide now, you're going to see the term endospore. So what is an endospore? So some prokaryotes can form endospores, and they do so during very harsh times. So there's like a lack of water, a lack of food. So during harsh conditions, the prokaryote kind of goes into a hibernation mode almost. And they form this very sturdy endospore. So it's the exact same bacteria forming the endospore and kind of hiding out, almost like a turtle withdrawing into its shell. So I don't want you to confuse a prokaryotic endospore with the sexual stage of plants, which is called a spore, or a fungal spore. So fungal spores and plant spores are reproductive structures, whereas an endospore is the same prokaryote kind of hibernating, going inside of its turtle shell. It actually forms this endospore until it can be found, it can be in good conditions again, then it will come out of the endospore. So that is on one of your terms on your first page. Most prokaryotes are going to reproduce by binary fission, and they can do this like every 20 minutes if the conditions are favorable, which basically means they just split into 2 cells. So your next 4 terms on the study guide are photoautotroph, chemoautotroph, photoheterotroph, and chemoheterotroph. So this is the nutritional diversity of prokaryotes. So we're going to look at both the energy source and the carbon source. So if the word has photo in it, for example, photoautotroph and photoheterotroph, you can think, what do I need to make a photograph? You're going to need light. So the energy source, if it has photo in it, is going to be sunlight. For chemoautotroph or chemoheterotroph, the organism is going to need chemicals, either inorganic chemicals or organic compounds. Now, when you look at the terms autotroph versus heterotroph, we've talked about autotrophs and heterotrophs before. So autotrophs are going to get their carbon source just from the air, from CO2. So whether it's a photoautotroph or a chemoautotroph, it's getting its carbon from carbon dioxide in the air. Heterotrophs, whether photohetero or chemohetero, are going to need to kind of eat somehow, so they have organic compounds. The next 2 slides also have these terms defined, or you can stop the video to rewind and get this. So photoautotrophs, photosynthetic, they include the cyanobacteria. Chemoautotrophs need carbon dioxide. They extract energy from inorganic substances. Now this is going to be like a multiple choice question on your exam. Here's photoheterotrophs. Again, if it's got photo in there, it's using light to generate ATP. It has to obtain carbon in organic form. Chemoheterotrophs must consume organic molecules for both energy and carbon. So let's move right along, and you can stop the video if you need to. So prokaryotes have a major impact on the Earth and its inhabitants. So let's talk about the different lifestyles of prokaryotes. So we have pathogens. This is your next term in your study guide. A pathogen are bacteria and other organisms that cause disease. So bacteria and other organisms that cause disease. And then the way that they cause disease is by producing poisons. So on your study guide, you've got the next two terms, exotoxins and endotoxins. So exo, like the name implies, is going to be poisonous proteins secreted by bacterial cells. Endotoxins are actually chemical components of the cell walls, and the only way these guys poison you is when the bacteria dies and it starts to break down, then those cell wall components are released to poison you. So anyone who's worked in the restaurant business knows the best defenses against bacterial disease. You have to have sanitation, antibiotics, and education. Washing your hands, just like we're being told to now during this coronavirus thing. There are two videos I want you to watch with this chapter. We've got the early life video available on Desire to Learn, and we've also got a video on antibiotics. If you get nothing else out of my class, I want you to realize that antibiotics only work against bacteria. Antibiotics do not do anything against viruses. I'm going to skip Lyme disease because that's a back east thing. It's not really something that occurs in Colorado. We have our own disease called Rocky Mountain spotted fever. So you definitely want to watch out if you go hiking and get ticks on you. All right, so we talked about prokaryotes as pathogens. Prokaryotes also play essential roles in the chemical cycles in the environment, breaking down dead stuff, poop. We're going to find this is very, very important in our next ecology unit. So moving down our study guide, we've got bioremediation as your next term. This is the use of organisms to remove pollutants from the water, air, and soil. Common thing in most cities is going to be sewage treatment, where you need to break down anything that has human waste in it. So they spray the water after the big chunks have been taken out. They spray it onto a rock bed, and then we have bacteria, archaea, and fungi that break it down. This outflow is then further purified before it's allowed to return to your home. But sometimes in the parks, they will use this outflow to water the grass. So this is much cheaper to use this outflow from this to water things like grass. So oftentimes you'll see a sign like, this water is not potable. So that means don't drink the water coming out of the sprinklers at some of our parks if there's a sign warning you not to do so, because it has not been purified, not tap water quality. Okay, so we've got certain bacteria that can help decompose petroleum, useful in cleaning up oil spills. There's a place in Golden, Colorado, the National Renewable Energy Lab. They used to work, I don't know if they still do, on projects like this. So now we're looking at, so how could eukaryotic cells have formed? So two things must have happened. So one, because prokaryotes are very simple and eukaryotes are more complicated, the eukaryotic cell's endomembrane system. So there must have been like a lot of folding in of the plasma membrane of a prokaryotic cell. And again, remember, we've got millions of years to do this. And then the second process had to be endosymbiosis. And the endosymbiosis is your last term on the study guide, and I realize it's not written very well here. But endosymbiosis is when a cell takes in another cell inside of it, does not eat it or break it down, but just kind of holds it captive. And you can think, well, that's not cool. You're having like these other tinier cells be slaves. But the tinier cells are also being protected by the bigger cell. So it's actually one cell living within another cell. The smaller cell likes the protection, and the bigger cell is getting energy from that smaller cell. And this is how eukaryotic cells evolved, because they have evidence that mitochondria have their own DNA. So why would they need that unless they used to be their own cell? So mitochondria living within a bigger cell, and then gradually they evolved together so that they just live together now as eukaryotic cells. This is showing that process. We've got an ancestral prokaryote that is simple, and then we've got membrane enfolding to make a nuclear membrane. We've got the endomembrane system, the Golgi apparatus, the smooth and rough endoplasmic reticulum. And then this shows either mitochondria, which were aerobic prokaryotes, living within that cell, or both, where we have chloroplasts and mitochondria living within one cell. So now the very last thing we need to talk about are going to be the 4 major categories of protists. So this is on the top page of your second page of the study guide. So let's talk about those. So the first kind are protozoans. So on your study guide, it says, what are the 4 major categories of protists? Give 2 examples from each category. If you just give 1 example, that's fine. So we've got protozoans. The easiest one to remember here is going to be, whoops, I went backwards. It's going to be an amoeba. Okay, so put protozoans, amoeba is the example. And then the second one is going to be a slime mold. So an example of a slime mold is a slime mold. Ooh, that's a tough one. Then the third type of protist is going to be a unicellular algae. So a single-celled algae. And an example of that, the easiest one, is a green algae. So unicellular, they're colonial. Just green algae is the easiest one there. And sorry I'm whipping through this so fast, but I'm getting you the answers to your study guide. Otherwise, we would have had to skip this chapter because of the week we had taken off. So our 4th kind of protist is a seaweed. So seaweeds are very different from the other kinds of protists because the other guys were like unicellular. These guys are large, multicellular. You might think that they're more closely related to plants, but they're actually not. So this is why they're in their own kingdom here. So this is pretty easy. The 3 major groups of seaweeds. We've got green algae, different from the unicellular green algae, obviously, but we've got green algae, red algae, and brown algae. So that should have gotten us through our chapter 15 study guide, so we did not have to skip this chapter. Sorry, there's no cool stories. If we look, here's another picture showing you how evolution could have happened from a unicellular eukaryote to a multicellular. So this is showing a unicellular protist that formed a colony, and then we've got specialization of cells, with some deciding to be motor cells, some are food synthesis, and then eventually, remember, we've got millions of years here, we can move to making some kind of sex cell. So that brings us to the end of chapter 15. I went super fast. Thanks, class.